So today we're going to start the second major division, I guess you could say, of immune, and that refers to the adaptive. So we're still going to talk about some of the cells we talked about previously, some of those activities, right? But we're going to layer in this adaptive component of your immune system that is continuously adapting and changing over your entire lifetime, right? Unlike your innate, right? All those things you're born with, they work all throughout your lifetime. They don't really change much, okay? This adapts and changes. So it develops the most effective means for eliminating an invader, right? So this is where we fine-tune it. The problem with this is that it can take a week or more to respond, right? Because that's how long it takes for it to recognize what it's been exposed to and produce the products to respond. In the meantime, right, our innate that we're born with must protect us. Those phagocytic cells, that inflammation, right, the phagocytic cells coming in and destroying. The sad news is that in some cases, the person may not survive long enough, right? So the innate might not be enough to take care of the invader, right? Adaptive hasn't come online enough to be able to protect you, and therefore you die. Adaptive, the great thing about this, in addition to being able to adapt and change over time to what we're exposed to, is it remembers what it's been exposed to. It has a memory component. So, you know, whenever you've done something the first time, right, you kind of fumble along, right? You're trying to work through things. And then the second time it goes smoother. The next time it goes even smoother. You get faster and faster the more you practice something. So your immune system is very similar in that aspect, that each time it gets exposed to something, its response is going to be that much faster, that much stronger. And that's because of the memory component. Vaccination takes advantage of your adaptive immune response. It's priming your system. It's giving you an introduction to the pathogen, to the antigen, to the danger signal at a safe level, right? Vaccination is at a safe level, or it's even been modified so it can't hurt you. Whether we're giving you a toxin that's been modified, whether we give you a virus that's been modified, whether we give you a bacteria that's been modified or killed, or pieces of something, whether it be a virus or bacteria. It is to let your system know what these things look like and develop memory of what they just saw. So that the first time you're exposed to it, naturally, other than vaccination, you don't have to wait that week or longer for your immune system to figure out what it is and respond. It should hopefully access its memory and go, oh, I've seen this one before, right? I know what to do. This response is at a molecular level, right? So you know how we talk about bacteria and viruses, but they're made up of molecules, right? They're made up of protein coats for viruses or envelopes with protein in them. You've got bacteria that have lots of different molecules on them, right? They have lipopolysaccharides if they're gram-negative. They have other molecules that are associated with them. So there's lots of different molecules that make up these organisms. So our response is literally down to the molecular level, particular proteins, sugars that are on their surface or make up these organisms. Does anyone remember the term we use for something foreign that our immune system responds to? Antigens. So we're going to look right even at the molecular level when we talk about these antigens, about these foreign substances. And then the other thing about our immune system has to be able to tell the difference between self and non-self, right? We don't want to attack ourselves. That's when we have autoimmune disease, right? When the immune system is attacking oneself, which unfortunately does happen for some people. But for the most part, we guard against this by this system learning the difference between self and non-self, right? What's a pathogen? And even when our own cells are behaving badly, right? Cancerous cells. To recognize healthy cells, healthy self cells from dangerous non-behaving cells like cancerous cells. So as you can imagine, this is quite a complicated system, right? Multiple layers to it. And so right away, too, we're going to kind of divide it up into two major aspects of adaptive. We're going to look at humoral immunity. Remember, he referred to the humerus, right? This human element, right? And another name for this that we use a lot of times is antibody immunity, because this is the most common product that you're going to produce in your humoral immunity. And the reason why it has this name, like I said, it was one of those things, this human element, right? This protein, this antibody that we're able to study very early on in studying the immune system. The cellular response, as the name implies, is cells, right? So this is cells attacking cells, right? This is when stuff gets inside of your cells. So when we talk about this, we talk about the different type of pathogens, and there's different sets of cells that are involved in these two different reactions. So remember from the first web pages that I sent you guys to, right? So this guy right here, this white blood cell, this lymphocyte, is saying, I recognize that one attack. And again, that's that memory component, right? That he has seen this before, right? And in this case, as I said, it's a molecular interaction. So notice, again, a lymphocyte, and it's binding. These are clearly the bad guys with these cute little crowns, right? And each one of them's got a different crown. So notice, he only recognizes one specific one, right? He doesn't recognize these other two bad guys. Their little crowns are a little bit different, right? His receptor on his surface, because this is a molecular interaction, is only recognizing this one bad guy, right? So it's down to that level of specificity, right? Very specific as to what they recognize. So I'm not gonna actually, I was gonna play this one, but I didn't set up my audio. So I'm gonna go ahead and audio it. And I had fast forwarded through it earlier. So I want to turn that off so it's not talking in my ears. So here we say have a cell in our body, and it's being attacked by a virus. Oh, actually, they're showing a macrophage. Sorry, forgot what part we were on. I'm going to say it, and it doesn't, I'm not even going to say the right thing. Okay, so pause. This is where I want to pause. So phagocytic cell, like a macrophage, can engulf something. That could have been a bacteria, could be a virus, anything foreign, right? It took it in. And remember I said there was one additional step that they were going to do after phagocytosis. Two cells can do this, macrophages and dendritic cells. They can do what's called antigen presentation, and that's what they're doing right here. So they have a special molecule that they can do this with. It's called MHC, and in this case, for this organism, this cell, it's class 2, okay? Antigen presenting cells have class 2, and we're going to go over this again. This is just the introduction. Right? So they're grabbing onto a piece of the antigen, right? So again, a molecular level. Other cells, right, have receptors on their surface for the antigens, and they're able to interact with this antigen presenting cell, right? So they match up. It's a helper T cell, as the name implies, he's helping out, right? They're going to communicate with each other. So this is interleukin 1, but what's the general term we give to these chemicals they use to communicate? It's a general term. Starts with a C. Need to start studying, y'all.
Cytokines.
Okay, cytokines, you got it, right? So this is an interleukin, right? They're communicating two white blood cells between each other. Right, he's like, I recognize this guy, yep. I think you need to respond, right? He releases another cytokine, interleukin 2. This will bind on receptors on this cell and other cells, and it's going to tell them to do a specific thing, right? Another interaction that can happen is between a B cell and here is a cytotoxic T cell. So here's the B cell. Notice that it has on its surface what we're going to talk about soon. These are antibodies. But it also has MHC class 2. It can also present antigen. And this is the one I thought was the first one. All right, so a cell, just any one of our cells, right, gets infected. Say with an intracellular bacteria or a virus. It can present on its surface antigens that get inside of it. Our cells are constantly, are actually even presenting their own self-antigens. But as long as those molecules are not modified, right, and they're in their normal state, the helper T cells aren't going to recognize them as being foreign or abnormal, right? In this case, something foreign got in, and they're presenting it outward, right? This is on MHC class 1. And this is found on all your nucleated cells, even those macrophages. So all your cells have this ability, for the most part, to present stuff from inside on their surface. So in this case, a cytotoxic T cell comes along. It can recognize this MHC class 1 with its T cell receptor. And in this case, if it recognizes this antigen, this means that this cell is in trouble, right? And it needs to be eliminated. And that's where it gets its name cytotoxic T cell, because it will actually kill this cell that it recognizes being dangerous. So it's going to eliminate it. Other cells that can recognize foreign substances are your B cells, right? And that's because they have a receptor right on their surface called an antibody or an immunoglobulin. These are proteins. And as you saw in that first little cartoon we looked at, they're specific for a specific antigen. One B cell only recognizes one specific little molecule on an antigen. When it binds to that antigen, it's actually going to bring this complex inward, and it's going to present it on MHC class 2. And that's one thing I don't like about this animation, is that it shows this cell interacting with the antibody. Somebody goofed, right? It actually is going to interact with the presenting of that antigen on this molecule. You see that there? They got it matching up in the wrong spot. They goofed. But once that cell is stimulated, one of the things it's going to do is going to make copies of itself, right? So it's going to what we call proliferate. It's going to produce a whole bunch of copies, because we don't want just one cell that recognizes that antigen. We want lots of cells. And then they're going to differentiate into plasma cells. Plasma cells actually secrete, as you can see, the antibodies are leaving the cells, right? And so these antibodies can leave the cells, they can go through your blood, through the fluid in your body, and they can interact with that specific antigen that started this whole chain reaction. So whether it be a bacteria or a virus, before it gets into our cells, these antibodies can bind to it and mark it for destruction. One of the things that can happen is a phagocytic cell could come along, and because it's coated with antibody, it can grab onto it. So antibodies in this case are opsonizing. They're tagging for destruction. But this isn't the only thing antibodies can do. So we've learned two other things that antibodies can do already. Or one. I think just one. I think just one for you guys. So there was a complement system. Do you remember that? The series of proteins in your blood and in your fluid. That system can be activated by antibodies, right? It can bind to this. That's the classical pathway. It can be activated. Do you guys remember the three major outcomes of complement activation. What does C3A and C5A cause? What's the most important immunological response? Inflammation. Inflammation. What does C3b have the ability to do?
Binds to bacteria.
Binds to bacterial cells, opsonizes them, just as this antibody is doing in this picture, right? And then sometimes we'll get the formation of the MAC complex. What does MAC stand for? What is it? Attack.
Complex.
Yeah, it's an attack complex, but what does the M stand for?
Membrane.
Membranes, right? It attacks membranes. It's that donut hole shaped group of proteins that can insert into membranes. So remember, who has a membrane as their outermost covering? Gram-negative or gram-positive? Gram-negative. So remember, this is very effective at killing gram-negative, as well as our own cells or human cells that don't match up, right, that we don't recognize. They could be killed by this system. So that once this is brought in, right, and then in addition to those cells that are going to proliferate, and some of them are going to differentiate into plasma cells, some are going to become memory cells. So these are long-lived cells, right, that have already recognized this pathogen before. So if we see it again, the response is that much quicker, right, because we already have these cells primed, waiting to see that antigen again. And that is just a little bit of a summary, right, of humoral and cellular. So we're going to go into more detail as we go. Right, primary response, that first time. And it remembers how it responded last time. Was it a cellular response? Was it humoral? Did we do both? And again, notice specific antigen. The second time we're going to respond so much faster. So for humoral immunity, where you saw us producing those antibodies, this is one of the most common ways that we're going to respond to extracellular antigens. So antigens that tend to stay outside of our cells, although not all bacteria do, right? Or I should say have a period outside of our cells. Let me put it that way. Okay. That's what they mean by extracellular. They can be found outside of our cells. So bacteria, toxins, viruses, there's always a period when they're outside of our cells that they could be traveling in our bloodstream. So our immune system, those phagocytic cells, will take it in, they'll process it, and they'll present it to B cells. And hopefully B cells will make antibodies against it. The cellular immunity is specifically against our own cells. So the only real invader that it helps the most against is viruses. But intracellular bacteria as well. And then cancerous cells, cells behaving badly, right? That's going to be recognized by our cellular immunity. Both of these are very powerful and can even damage our own tissues if misdirected, right? So if it gets the wrong signals, right, and this is why you'll notice there's lots of communication. They didn't just hook up with each other via those receptors, they also sent chemical signals, right? So they communicate multiple ways about what's going on. So that's what they mean by verification is usually required. Yeah, so he asked if anaphylaxis. What anaphylaxis is an immunological response, but one that's damaging to the body, right? And so that's usually referred to when you're exposed to something that isn't necessarily going to harm you. Anaphylaxis can happen with allergy, but it also can happen with certain toxins. So you could say someone's in anaphylactic shock. And that's when their immune system, again, is responding to this antigen very aggressively. So much so that you have so much inflammation widespread that you could go into shock. And when you have lots of inflammation widespread, you get that drop in blood pressure. And that's what they mean by shock. When someone's in shock, their blood pressure drops dramatically, and they don't get perfusion to their vital organs of blood. You don't get enough blood to where it needs to go. And that's why it can be deadly.
Is it true that you become allergic to anything at any time? Even yourself?
Yeah, you could, not that you would, right? So you can he said you can become allergic to anything at any time, and this is true. Some people are more predisposed to allergy than others, and it has been linked to hereditary. But any one person could become allergic at any time in their lifetime. And you could be even allergic to yourself. And that's what we term autoimmune disease, right? When the immune system is attacking self. We use that term instead of saying being allergic to yourself. Sounds kind of weird. So for these responses, your body sometimes will go kind of all in one direction, where you just have an antibody response. Sometimes it'll go all in a cellular direction, or sometimes you'll have a mixture of both cellular and humoral immune responses. So our major player from our innate immunity is a dendritic cell or a macrophage. Remember, these are the ones that are going to be able to grab on, digest by phagocytosis, but then they go one step further and present. And that's really what a dendritic cell specializes in, and that's why they show him as the main player instead of the macrophage. Because the dendritic cells, that's really their main job, is to share this information with the lymphocytes, right? So your lymphocytes are your T cells, right, and your natural killer cells, which we'll talk about later, and your B cells. Remember those three lymphocytes. So naive means that they've never seen a bad guy, right? They just haven't seen it yet. They're naive to the fact that the world is bad, right? But then a dendritic cell is going to come along and he is going to present it. And remember, he's going to be presenting it molecularly. He's got a receptor, he's picked up this piece and he's presenting it. They're not showing you that level of detail in this diagram. And he can present to either type of T cell, whether it be a helper T cell or a cytotoxic T cell. Helper T cell, as the name implies, are going to help out other cells. So they'll help out macrophages. Remember, sometimes they need a little bit of an extra push in killing power, right? They're not very aggressive killers in the beginning of an immune response. Maybe if the helper T cell signals to them, hey, I recognize this one, this is really a bad guy, you really need to amp up your killing ability, they can get them to do that. Then they can also help out B cells. Remember, B cells have receptors on their surface for antigen all by itself. So maybe they've come across an antigen and they need that verification signal, that extra signal before they move on, right? Then the helper T cell can help with that verification, that they've seen it presented from the dendritic cell. Yep, it's a bad guy. And then the cells go forward to proliferating, and the most common cell type they're going to differentiate into is those plasma cells. And what they're showing you here is that their endoplasmic reticulum, their rough endoplasmic reticulum, gets really big. And that's because they're going to secrete proteins. Antibodies are proteins, right? And that's what the rough ER makes, right, is secreted proteins. So here are the antibodies, they're secreted, and then they can go and bind directly to the antigens, and we're going to talk about then once that happens, how our immune system recognizes and clears away these things. What benefit does antibodies binding have, right? And most commonly, tagging for destruction, right? So that what we call opsonization. The same thing that C3b can do, so can antibodies. But antibodies is molecularly specific, right? Where C3b is general, right? Sticks to most bacteria. This is going to be very specific to a bacteria or a virus or some other pathogen. Now, this other subset of T cells is your cytotoxic T cells. And so this is when a cell, say, is infected by a virus, right? And it's presenting those viral antigens on its surface that this dendritic cell has already recognized earlier and told the cytotoxic T cell about it, so it proliferated. And now these guys are going out literally hunting for cells that are virus infected, and they're going to kill them, right? Whole bunch of them once it's been activated. So with a virus response, we could get this cytotoxic response, but we can also get antibodies produced. Because when the viruses are outside of our cells, the antibodies can bind to them, and it essentially blocks them from binding to our cells. Because the very first thing for infection is attachment, right? For bacterial cells, they have to be able to stick to us. For viruses, they have to attach and then get into our cells. If we can stop that very first thing of attachment by blocking them with antibodies, right, infection can't happen of that cell or even of our body. So, as I mentioned, these lymphocytes, B cells and T cells, are specific for specific antigens. They have receptors on them specific for antigens. There's a distinctive difference here, though. Notice that the B cell, as I said, their receptor is actually an antibody, what we call an antibody. So it has 2 antigen binding sites on it. And they're showing the antigen binding there. The free antigen all by itself can bind to a B cell. Unlike T cells have to have antigen presented, whether it be by an antigen presenting cell like a macrophage or a dendritic cell, or one of our own cells that's maybe infected with a virus or a bacteria. So they have that T cell receptor that's specific for the antigen. In addition, they have another molecular that we refer to as the CD marker. These are referred to as clusters of differentiation because we can actually differentiate the 2 different types of T cells based on which CD4 marker they have. So what 2 types of T cells did I mention so far? Helper T cells, and who else? Cytotoxic T cells. Helper T cells are also sometimes referred to as CD4 cells, the number 4, because they have this marker CD4 on their surface. And it allows them to communicate with antigen presenting cells. So how I remember this one is, you ever hear 4-H, right? 4-H. Right? So again, here, 4 for CD4, H for helper, right? 4-H teaches kids how to help and learn about animals, all that good stuff. So 4-H, remember 4-H, 4 CD4, H for helper in this case. The other one is CD8. And 8 sounds like to eat, right? Eat, 8, the number 8 sounds like eat. And what do these cells do? They eat or kill other cells, do they not? Cytotoxic CD8. I always think 8, eat. That's how I distinguish the 2 of them. So hopefully that helps you guys out. So how and where do all these interactions happen in our body? We have specially designed vessels in our body that help carry components of our immune system. Those are our lymphatic vessels. There are certain areas and sites in the body where these interactions happen between these cells. These are your secondary lymphoid organs and tissues. And then the primary lymphoid organs is where your white blood cells are born and where they're educated, where they learn the difference between self and non-self. So the lymphoid system, right, is a collection of tissues and organs designed to bring those T cells into contact with the antigens, right? The dendritic cells travel there with the bad stuff. Right? The superhighway that they commonly travel on, other than the bloodstream, is our lymphatic vessels. Right? These organs where this interaction happens is the secondary, and the primary is actually where your lymphocytes are born and educated. So, there's only two primary lymphoid organs. Where are all your white blood cells actually born? On this diagram, where are they born? In the bone marrow, right? They're born in the bone marrow. And then they're going to leave the bone marrow, and for B cells, they're mature. So remember, B, bone marrow, done, right? T cells, on the other hand, have to travel to one of these organs, and they're named T cells because this organ starts with the letter T, the thymus. And that's where they learn the difference between self and non-self. The T cells, right, are the ones that have really got to recognize self from non-self. Okay? They're the ones with those CD markers, right? CD4 and CD8. And then I've got to recognize MHC class 1 and 2. B cells, when they leave the bone marrow, they have their B cell receptor, and they can go out and they don't have to interact with cells, right? They got to be able to interact with antigen all by itself. They can interact with cells too, but they're not going to be telling the difference between self and non-self. That's your T cells. So all the rest on this list is what we call our secondary lymphoid tissues and organs. Okay, so your lymph nodes, which are represented by these green dots all throughout, are little filters for the lymph, which is the fluid that travels through your lymphatic vessels, all these green vessels shown on this body. This is not circulation, this is lymphatic vessels. And these start in your tissues, and they drain as they go through several lymph nodes. Eventually that fluid will enter back into circulation because that's where it came from, right? Because remember, when we have inflammation, the white blood cells come out of the blood, and so does some of the fluid, right? And that's why you get the swelling. So that fluid, if that was to just go right back into your bloodstream, it could contain pathogens, viruses, bacteria, right? Things like that. Do you want that going into circulation and then spread all throughout your body? No, that would be bad, right? It's bad when pathogens get into your blood because that's the superhighway, right? That's going super fast. Right? It's going to go, you can go from one area of your body to the other in a minute, and this is at resting heart rate, right? You're just sitting here right now. You get a free ride in as little as a minute from one section of your body to the completely other side. Okay? So we keep that guarded. So instead, to get that fluid that we need back into circulation, it gets soaked up by the lymphatic vessels. And along the way, we're going to filter it through the lymph nodes. Right? In the lymph nodes are macrophages and dendritic cells and T cells and B cells, all of this to be able to gobble up and respond to any pathogens that are in that lymph. And so that hopefully by the time it gets up here to your subclavian veins, right, it has been filtered enough that no pathogens are going into your bloodstream. But we have one protective organ. Its main job is to protect us for anything getting into our blood and filters it out. Anyone know which one on that list has the job? Your spleen. Your spleen captures pathogens in your blood. It is the one organ you can lose and live without, but you really wouldn't want to, right? You have to really worry about bloodborne pathogens. The other functions that your spleen does, like red blood cell recycling, your liver will take over, right? But you can live without your spleen, although I wouldn't choose not to have it, right? It does help you out. It's a very important secondary lymphoid organ in that it's filtering your blood, continuously filtering your blood. And there are macrophages and dendritic cells and T cells and B cells right in that spleen to respond right then to anything that it pulls out of your blood. your tonsils, and you have several tonsils. You have one that's associated with your tongue, referred to as a lingual tonsil. You have your palatine tonsils, which is probably what they're showing you here in your throat. And honestly, you really don't see those unless they're really inflamed and you're looking down someone's throat. That little punch-in bag that you see when you look in that's part of your soft palate. It's referred to as your uvula, right? Your soft palate is to keep from when you swallow stuff going up your nose, right? Because your nose, your nasopharynx, the part of your throat that's connected to your nose, is right up from there, right? So stuff drains down that part of your throat, right, into your mouth, down the back, right, and then into the laryngeal pharynx, which is the part of your throat associated where your larynx is, where your vocal cords and things such as that. So those can become quite swollen. Sometimes some people have had them removed, right? Because they keep flaring up. The unfortunate thing is they're in your throat, right? So they make it difficult to breathe, make it difficult for you to eat, swallow, things such as that. And then you have another tonsil. It's referred to as your adenoids commonly, which is plural, right? It's only one tonsil though. It's your nasopharyngeal tonsil, and it's found in the back of your nose, right? In the nasopharynx, so the throat portion, but up in your nose region. And this one, unfortunately, where it's located too is in between the two eustachiary tubes, which are what drain your middle ear, your inner ear. And so, like for some young children, sometimes that will swell, right? In the case for my son, in addition to having tubes put in his ears, before we found out it was allergy causing fluid to build up in his ears, they removed about 80% of his adenoid because it kept swelling up too big, that it would block his eustachiary tubes and keep them from draining. So this is a common thing things that can sometimes happen for people. I almost wish, that mine was noticed when I was younger, because probably would help some of the inflammation I deal with in my nose if I didn't have most of my adenoids. But they're located there at a particular strategic place, right? This is where a lot of stuff enters into your body, right? And so right there, you've got dendritic cells, macrophages, T cells, B cells that can respond very quickly to the stuff that you're inhaling. Your appendix, which is also a tiny little sac that hangs off your cecum, which is a portion of your large intestines or your colon. This unfortunately has a lot of white blood cells in it, and if stuff gets compacted in there, it can get inflamed. The bad news with that is if it gets inflamed and bursts, then you have your intestines leaking into your abdominal cavity, right? And that can be life-threatening. Why we have that collection there, one only knows. And then, so that's mostly it for your organs, but you have mucus, right, that lines your entire reproductive and respiratory and digestive tract. Because remember, these are all openings as well that pathogens could use to get into your body. So in addition to the mucus that helps bathe, under that mucus layer are collections of these white blood cells. So we refer to this as mucus-associated lymphoid tissue. Lymphoid because it's got those B cells and T cells and the dendritic cells and the macrophages to be able to respond right there. And they actually even produce a special class of antibody that gets secreted into your mucus. It's called IgA. And we're going to go over the different classes later when we go over the antibodies. And then your skin. Remember, that's a very large protective barrier. Under your skin, within your skin, there are regions where you have concentrations of these lymphatic cells. We refer to this as skin-associated lymphoid tissue, right? And so SALT for short, which is kind of funny because the skin is kind of salty sometimes too, which has antimicrobial ability as well.
Would like Langerhans cells be one of those?
I don't know the specific names that they give to some of the cells. It might be.
I think that one was for antibacterial.
Very well could be. So these secondary is where these interactions are happening. So this is, it gives you an example of your gut, right? Antigens are coming in, and you actually, even in your gut, you have specialized cells that are always continually sampling stuff that's coming through your intestines, right? These are your M cells. And then look at the dendritic cell. He can even stretch his little dendrites, his projections in between the cells of the epithelium of your intestines and grab pathogens and bring them in. All right, the M cell is going to present them to the macrophage. Remember, both dendritic cells and macrophages can digest and present. And so then you're going to have regions. So this region that in your gut is referred to as a Peyer's patch, right? That's a particular name to your mucus-associated lymphoid tissue that exists in your gut, in your intestines. And notice there are lymphatic vessels, right, that they're going to drain any fluid. And they actually work in your intestines dual purpose. They actually help bring in fatty substances. The fat actually travels through your lymph because fat isn't very water soluble, so it doesn't travel through your blood. And most of your fat soluble vitamins, of course, too, go through there. But notice that there's an area right outside the macrophages and dendritic cells of T cells denoted in blue here. And then B cells here. And the B cells tend to form these circular clusters because once they start proliferating, right, they form this big, huge mass of cells. And there's a similar architecture in your lymph nodes too. It's directional. The lymph comes in one end, gets exposed to the macrophages and the dendritic cells, and they're going to present to the T cells and the B cells. So as I said, it's an ended system. It starts, your lymphatic vessels actually start. They're blunt-ended. They start in your tissues. Okay? And then they gather all up together and form two ducts that dump all the lymph into your two subclavian veins, your right and your left. But all along the way, it's going to get filtered by lymph nodes. And then if you'll notice, and I'll flip back to this other, so here is your blood system, right? And if you have inflammation, that extra fluid doesn't go back in. It's going to get absorbed by the lymphatic vessels. So notice here, right? You have lots of collections in your neck region and your armpit region. You have a lot in your abdominal region, right? Because there's a lot of stuff that could get through your intestines, right? And you want to be able to drain that. You have large collections, right, in the mid area, you could say, of your body. Not to say you don't have them along your arms and stuff like that, but you definitely have a greater collection as you get inward. And so this is why sometimes too, like you'll have swollen, they'll say you have swollen glands. They're referring to your lymph nodes in your neck, sometimes when you're sick, right? And that's because they're responding to infection, okay? Sometimes some people might even feel them in their armpit region or groin region, depending on what type of inflammation you're having. But the most common where they really show up and you can feel them is usually in your neck region, right? And they'll be sore and tender, right? And the doctor will palpate them and you'll want to smack them, and that's a good thing. When they're hard and they're not tender, sometimes it could be that they're cancerous, right? So you actually want them to kind of be tender. You don't want them to be cancer. Not to say if they're not tender that they are, right? Just if you've got swollen lymph nodes, you should always get them checked out, right? So the next thing that I want to talk about, and I was right that I wasn't going to get to the different antibody activity, but that antibody activity that's posted on Blackboard, I do have printouts for you guys to be able to write right on as we go through that next time. I am going to cover this last objective, though. It's short. Okay, so remember we talked about antigens, right? And that this is a molecular interaction. So when we say a bacteria, like E. coli, E. coli 0157, that's a particular strain of E. coli. It has that molecules, those sugar molecules, that we denote 157 on their surface. And one of the ways we're able to easily detect this is that we have antibodies that specifically recognize that antigen, that very small molecular structure that's different on that E. coli from any other E. coli, right? So when we're talking down to the molecular level, what antibodies are actually binding to, these are referred to as epitopes or antigenic determinants, right? So we're going to look at a picture of that. So say this is a protein, right? And down to the molecular level, we're talking about lots of different amino acids here, right? And the amino acids in this region, these all vary, right? They're going to be different, typically. So this is 3 different spots, right, that would be accessible by an antibody that we could bind to. But what kind of antibody do we need to bind? A different one, right? The antibody that binds to epitope 1, will it be able to bind to 2? No, because that's different molecular structures. So your body, when you are exposed to something even as small as this protein, an antigen as small as a protein, you don't produce just one antibody that binds just at one site. You'll produce multiple antibodies against the same antigen, right, that bind at different molecular sites, right? We refer this as polyclonal. Poly meaning many, clonal meaning that that antibody was only made from one set of B cells that were all the same. Clones are all the same, right? So as I said, this is a very specific interaction of that antibody with the antigen. So look at something a little bit larger. Here's a bacterial cell. So like I said, that E. coli 157, maybe that's the O157. This one antibody binds to it. So what scientists have done is they've taken out and have been able to purify that one antibody. So they can use it for testing to detect that one specific strain of E. coli, right? Because it's down to that level of molecular recognition. It's that specific. That antibody-antigen reaction. So for a bacterial cell, they may make it against the O157. This may be against some other group of molecular markers that are found on its surface. This one's a different color denoting in a different shape, letting you know that this antibody is binding to something different on that same bacteria. Does that make sense to you guys? Right? So as I said, write down very specific to the molecular level these interactions between an antigen and an antibody. And when we say antigen, right, we're talking about something foreign that the body is responding to. Right? And how it got its name, antigen, right, is the most common thing we make against an antigen is an antibody. So antigen stands for antibody generators. Right? But nowadays, anything that elicits any type of immune response, because we didn't just talk about antibodies today, right? Cells can respond, right? Cells can be cytotoxic and kill other cells. That's an immunological response, is it not? Yeah. So an antigen, in general terms nowadays, is just anything foreign that elicits an immune response, right? Whether it be antibodies or also a cytotoxic response, right? A cellular response, right? And as you saw, proteins, polysaccharides, we're talking down to molecular level. Those are really good antigens. Nucleic acids and lipids, not so much. And this is only because they don't vary very much, right? They're pretty consistent molecular structures. They don't change very much. All right, that's the problem with them. Where proteins, right, we've got 20 different amino acids. We get gazillions of different combinations and arrangements, right? And so we can be very specific in our interactions. Okay? So next time we're going to actually look at the structure of an antibody, what binds, and then we'll talk about once the antibodies bind, how our body responds to that, right? What are the outcomes? Why are antibodies so awesome and protective, and probably one of my favorite molecules. All right, see you guys on Wednesday.